Hi everyone, welcome back to my channel. Today I would like to continue my lectures on double fertilizations. So, life cycle of angiosperm. The life cycle of angiosperm start with sexual reproduction, occur in flowering plant in specialized structures called flowers. So in the flower structures, we can see the stamen will produce pollen. So the counterpart of the stamen is pistil, which composed of stigma, the style, and the ovary. So the ovary is the place where it's produced egg. Sexual reproduction in plant involve the pollination, which is the transfer of the pollen from the anther to the flower to the stigma of the same or different flowers. So the pollen land on the stigma and grow into the style until it penetrates the ovules and release two sperm. So after the release of two sperm, the occurrence of the double fertilizations. So the double fertilization is a unique process in flowering plant. One sperm will fuse to the egg to form the zygote, and another sperm fuse to the pollen nucleus to form the primary endosperm nucleus. And after the fertilizations, it's followed with seed and fruits development, then seed and fruit disseminations, and then seed germinations again. This will create a cycle of angiosperm. Let's look at the male organ of the flower, which also known as angiosperm. So as mentioned earlier, the angiosperm is the walls of a stamen, which each stamen consists of an anther at the end of the filaments. The anther usually is made up of 4 elongated lobes called pollen sac. You can see from the picture here. Early in the development of anther, each pollen sac contains a mass of dividing cell called microsporocytes. Each microsporocytes divide by mitosis to form 4 haploid microcytes. The nucleus of each microspore then divide by mitosis to form a 2-cell pollen grain, which contain a tube cell and a smaller generative cell. The rules of these 2-cell haploid male gametocyte is to produce sperm cell for fertilizations. After the pollen grain are matured, the anther wall split open and the pollen is shelled. Pollen grain are transported to the stigma or adjacent or distant flower. This process is called pollination. The dry pollen grain then absorb water from the stigma and become hydrate. It also secretes protein, including some of that are involved in pollen recognitions and compatibility reactions with the cell of the stigma. This recognition mechanism is important because of it exhibits pollen from germinating and growing on the wrong species. These proteins are the same one that cause allergic and hair fevers. So let's look at the development of pollen from microsporocyte to pollen grain. Picture is adapted from the textbook. So we can see that the pollen grain is surrounded by an elaborated cell wall. The pattern on this wall is genetically fixed and it varies widely among major group of the plant. The wall contains sporopollenin, a very hard material that resists decay. As a result, this pollen grain make a good fossil, and botanists have found them useful in studying the evolutionary history of the seed plant. So look at the pollen gland that after germination it will form a pollen tube, shown in figure B. The pollen tube grows between cells in the center of the style, a region called the transmitting tissue. In some plants, such as the lily, the style is hollow and the transmitting tissue is reduced to a layer of secretory cell on its inner surface. The cells secrete a special material in which the pollen tube grows. In solid transmitting tissue, the pollen tube secretes digestive enzyme that partially break down the transmitting tissue cell, making it easier for the pollen tube to grow. The pollen tube grows by elongation very near its tips. It is filled with many small vesicles containing cell wall building material needed for the tube to grow. Endoplasmic reticulum and other organelles also are presented. This is a scanning electron microscope image which show 500 times magnifications of the pollen gland from variety of common plants such as the sunflower, morning glory, hollyhocks, and lily. So as you can see here, they contain variety of shape, colors, and the size is range from 15 to 200 micrometers. So now let's look at the female organs of the flower, which called gymnasium. So in its simple form, the gymnasium consists of a single folded scalpel or simple pistil in which the ovary reside. In more complex flowers, the gymnasiums may consist of several separate scalpels or several groups of few scalpels. A group of few scalpels is called a compound pistil, which such as shown in these pictures. The ovary is a hollow structure having from one to several chambers or locus. The number of scalpels in a compound pistil is generally related to the number of stigma, the number of locus, and the number of size on the ovary. Example is the pea plant have a single stigma and its ovary has one locus, shown in the first picture, the green bean, while the tulips, which is the picture at the left hand side and the down, the tulips pistil have 3 stigma. Its ovary has 3 sides and it contains 3 locus and 3 scalpel. So the tissue within the ovary to which an ovule is attached is called the placenta, which is the picture at the right hand side that you see. The manner in which placenta are distributed in the ovary is termed placentation. When the placenta are on the ovary wall, as in breeding hearts, the placenta is parietal. When they are raised at the exits of the ovary that have several locus, as in fuchsia plant, the placentation is axial. Less frequently, the ovary form on a central column that will be known as central placentation. So emanating from the ovary is the style, a stalk whose length and shape vary by species, with a stigma at its upper end. So often the surface of the stigma is covered with short hair that aid in holding the pollen grain, and sometimes they secrete a sticky fluid that stimulates pollen tube to grow or to germinate. In many windy pollinate plants such as the grasses, the stigma are elaborately branches or feather-like. So as you can see here, within the ovary is the ovule, the structures that after fertilization will eventually become the seed. As the ovule matures, it forms one or two outer protective layers, the integument. The integument do not fuse, leaving the small opening called microphylla. At the same time, one of the internal dividing cells of the ovule, the megasporocyte, is enlarging in preparations for meiosis. Let's have a closer look of the embryo sac. Is the female gametophyte plant. So after meiosis of megasporocyte, a row of 4 cells called megaspore is produced in the nucleus. Each megaspore is haploid. As a rule, the 3 cells nearest the microphylla disintegrated and disappear, whereas the one furthest from the microphylla enlarged greatly. This megaspore developed into the mature embryo sac in several stages. First, a series of T mitotic divisions occur to form an egg nucleus embryo sac, which show in the number 2. The nucleus migrate, and then thirdly, the cell wall form around the nucleus. At the end of this process, an egg cell and 2 synergic cells are positioned at the microphylla end of the embryo sac. Because it is frequently difficult to differentiate the cell from the other 2 cells, all 3 cells are sometimes referred to as the egg apparatus. The 2 nucleus that migrate towards the center are polar nucleus. They lie in the center of the large center cell. The 3 nucleus at the end of the embryo sac opposite the microphylla form 3 antipolar cells. During megasporogenesis, the diploid megaspore mother cell undergo meiosis and give rise to 4 haploid nucleus. Angiosperms exhibit 3 main patterns of megasporogenesis, referred to as monosporic, bisporic, and tetrasporic. These 3 patterns are summarized in this figure 2. The 3 types differ mainly in whether cell plate formations occur after this division, thus determine the number of meiotic products that contribute to the formations of the mature female gametophyte. In the monosporic pattern, both meiotic divisions are accompanied by cell plate formation, resulting in 4 one-nuclear megaspore. Subsequently, 3 megaspore, generally the microphylla, most megaspore, undergo cell death. In the bisporic pattern, cell plate form after meiosis 1 but not meiosis 2. So the result is two nucleus megaspore, one of which degenerates. In the tetrasporic pattern, cell plates fail to form after both meiotic divisions, resulting in 1 4-nucleus megaspore. Thus, these 3 patterns give rise to a single functional megaspore that contain 1 monosporic, 2 bisporic, or 4 tetrasporic mitotic nucleus. The monophoric pattern is the most common form and is represented within the polygonium pattern. So, double fertilizations. So after landing on a respective stigma, a pollen grain germinate and produce a pollen tube that extend down between the cell of the style toward the ovary. The pollen tube then will discharge 2 sperm into the embryo sac. In double fertilization, So one sperm will fertilize the egg and another sperm combine with the polar nucleus, giving rise to the food storing endosperm. So this is overall diagram show the double fertilization. The further explanations will be on coming chapter, embryogenesis. So when compare the seed and spore, we find out that seed is having more advantage compared to spore, such as it has a multicellular layer of tissue, while spore is only single cell, and it also have a very strong protective coat that prevent desiccations and also damage of the embryo. So it has a very big store food supply to supply food for the growth of embryo, and therefore it ensures the survival of the seeding plant in more extreme environmental or even fire, giving them a selective advantage over spore plant. So this table show what is the differences of spore compared to the seed. Basically, the size of seed is bigger compared to the spore, and there are two types of the spore, which is heterosporous and homosporous, while for the seed they have diploid or haploid. So of course seed is more complex compared to the spore, and the location of the spore is underneath the leaf area of the fungi, fern and moss, and for the seed is located in the fruits and flower. So the germination requirement for the spore is wet environment, and for the seed is everywhere.